All right, welcome to the second part of the Module 10 Cellular Respiration lecture video. In this video, we are going to look more in depth at the specific stages of cellular respiration. Cellular respiration occurs in three main stages. The first stage is called glycolysis. Glycolysis actually occurs in the cytosol or the cytoplasm. It does not occur in the mitochondria. The second stage is kind of a combo of pyruvate oxidation and the citric acid cycle. We're going to talk about those two together. Stage 2 does take place within the mitochondria. The mitochondria is denoted by this peach-colored bean-like shape. And then stage 3 is oxidative phosphorylation, which includes electron transport chain and chemiosmosis. That also takes place in the mitochondria. In stage 1, glycolysis, which will be represented by blue throughout this lecture, it occurs in the cytosol and it begins officially cellular respiration. Glycolysis breaks down glucose into two molecules of a 3-carbon compound called pyruvate. So we start out with glucose at the beginning of glycolysis. At the end of glycolysis, pyruvate is produced and a little bit of ATP is produced in the process. Notice that the amount of ATP that is produced is indicated by the size of the little bubbles. Glycolysis and then the citric acid cycle produce small amounts of ATP. The most ATP is produced over here in oxidative phosphorylation, since that little bubble is a lot larger. The second stage includes pyruvate oxidation and the citric acid cycle. Both of these take place within the actual mitochondria, and they're going to complete the breakdown of glucose to carbon dioxide. They also supply the third stage of respiration with electrons, indicated by these arrows right here. Pyruvate oxidation and the citric acid cycle, you can see that carbon dioxide is given off at this point. So this is where the carbon dioxide that's a byproduct of cellular respiration comes from. The citric acid cycle does also produce a small amount of ATP. And then glycolysis, pyruvate oxidation, and the citric acid cycle are going to funnel electrons that are carried by either NADH, which we talked about in the last video for this module, or FADH2, which is another electron carrier we haven't talked about yet. Those two electron carriers will shuttle electrons to stage 3, which is oxidative phosphorylation, where the electron transport chain and chemiosmosis occur. In the third stage, oxidative phosphorylation involves electron transport, the electron transport chain, and chemiosmosis. In stage 3, NADH and a related electron carrier, FADH2, shuttle electrons to electron transport chains, which are embedded in the inner mitochondrial membrane. Most of the ATP produced by cellular respiration is generated by stage 3 by oxidative phosphorylation, as evidenced by this larger squiggly thing around the ATP from oxidative phosphorylation. The electrons in the electron transport chain are finally passed to oxygen, which becomes reduced to H2O, which we can see right here, oxygen becoming reduced to H2O. Remember that acronym oil rig? Oxidation is loss, reduction is gain. Just remember those terms as we move through it. So if the oxygen is reduced to H2O, that means that oxygen gained electrons. Here's a bigger overall summary. Remember that mitochondria are found in both animal and plant cells, so cellular respiration occurs in both animal and plant cells and in the cells of most eukaryotes. We're going to go ahead and move to a checkpoint question. Of the three main stages of cellular respiration, which one does not take place in the mitochondria? Hopefully you answered glycolysis for this one. Stage 1, glycolysis occurs in the cytosol or the cytoplasm, not the mitochondria. We can see right here, glycolysis is occurring outside of the mitochondria. We're going to talk more specifically about glycolysis now, which is stage 1 of cellular respiration. In stage 1, glycolysis harvests chemical energy by oxidizing glucose to pyruvate. So remember, oxidation is loss. So glucose is going to lose some electrons. An overall summary of glycolysis is shown here in this image on the right. Glucose is represented here. Glucose is a 6-carbon molecule, so each one of those gray dots represents a carbon. You can see that we have 1, 2, 3, 4, 5, 6 carbons. Throughout the process of glycolysis, we see that glucose is oxidized and NAD plus is reduced. Remember, reduction is gain. NAD plus gains 2 electrons throughout this process to become 2 NADH, and then 2 hydrogen ions are produced as well. Throughout glycolysis, 2 ADP molecules are also phosphorylated back into ATP. The result of all of the steps of glycolysis are 2 pyruvate molecules. Pyruvate is a 3-carbon molecule, so we see that the glucose, which was a 6-carbon molecule, has been split up into 2 3-carbon molecules. In glycolysis, ATP is used to prime a glucose molecule, which eventually is split in 2. Those 3-carbon intermediates will then be oxidized to 2 molecules of pyruvate, which yields a net gain of 2 ATP and 2 NADH. This is the written out explanation of what happens during glycolysis. I think it'll be a little easier to look at this step by step. So that's what we're going to do across the next few slides. A reminder, glycolysis does not occur in the mitochondria. It occurs in the cytoplasm. Glycolysis is when glucose, which is a 6-carbon molecule, is split up into 2 3-carbon molecules called pyruvates, and 2 NADHs are produced and 2 molecules of ATP are produced. Those molecules of ATP are formed by substrate level phosphorylation, in which a phosphate group is transferred from an organic molecule to ADP. This is facilitated by an enzyme. So pretty much what's happening, an enzyme binds ADP and a substrate molecule with 2 phosphates. One of those phosphates is then transferred to ADP to form ATP, or adenosine triphosphate, and then the enzyme lets the product and the substrate lets them go. Sorry, I misspoke there. The enzyme will then let the products go. The substrates are over here on the left. So after ATP has been phosphorylated, the enzyme will release them. And this is called substrate level phosphorylation. A quick checkpoint question here. So for each glucose molecule processed, what are the net molecular products of glycolysis? This figure here will give you the answer that you need. So we have 1 molecule of glucose going through glycolysis. We know that it gives us 2 ATP. We know that it gives us 2 pyruvate, and we know that it gives us 2 NADH. So the answer is 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH for every molecule of glucose that goes into glycolysis. There are multiple reactions in glycolysis that actually split glucose into 2 molecules. I know there's a lot of text on this slide, but I wanted to kind of talk through what's happening, and then we'll go through it visually. So the last few slides really simplified what's going on in glycolysis. They had the main points included, but you couldn't see all the reactions that led to that point. Glycolysis is actually a 9-step process, so we're going to take a closer look at each of those 9 steps now. The following figures will show simplified structures for the organic compounds in the 9 chemical reactions of glycolysis. You can see some of the simplified structures over here on the right. The sequential steps of glycolysis illustrate how, in a metabolic pathway, each chemical step feeds into the next. In other words, the product of one reaction serves as a reactant for the next, which we can see happening right here. The product of step 1 is this molecule here, which serves as the reactant for step 2, and so on and so forth. So the product of one step is going to be your reactant in the next step. Compounds that form between an initial reactant and a final product are known as intermediates. This is our initial reactant here, glucose. Our final product is not shown in this image, but each one of these carbon molecules here would be considered intermediate molecules because they are formed in between the initial reactant and the final product. We can split glycolysis into two phases. The first phase is the energy investment phase. The energy investment phase actually consumes energy, so ATP is used in this phase. 2 molecules of ATP are used to add a phosphate group to each glucose molecule. So right here we have steps 1 through 3. Glucose is energized using ATP. Glucose is then split into 2 small sugars, which happens down here. We're going to walk through this step by step. So we start out with a 6 carbon molecule glucose. ATP is used to then add a phosphate group onto the end of glucose. So we still have a 6 carbon molecule, but now with a phosphate group. Again, we have our 6 carbon molecule with the phosphate group. ATP is used again to add a phosphate group to the other side of this molecule. So now we have a 6 carbon intermediate with a phosphate group on either end. That 6 carbon intermediate is then going to split into 2 3 carbon intermediates, which we see here. Each 3 carbon intermediate does still have an attached phosphate group. So these are steps 1, 2, 3, and 4. If we take a closer look at this is the same image just blown up. You can see that we start out with glucose here. Glucose becomes glucose 6-phosphate right here. Another chemical reaction turns that into fructose 6-phosphate, still with the same amount of carbons. And then a phosphate is added to the other end to form fructose 1,6-biphosphate. And then fructose 1,6-biphosphate is split into glyceraldehyde 3-phosphate, and there are 2 of them. The short term for that is G3P. I'll be transparent right now. I don't expect you to memorize all these intermediate compounds. I care most about the initial reactants in each step and then the final products. So if we're talking about glycolysis, I want you to know what the initial reactant is. It's glucose. And I also want you to know what the final product is, but you don't need to memorize all these intermediates. I am just showing you how complicated this process actually is and what is going on within the cells when glycolysis is occurring. So at the end of step 4, we have these 2 3 carbon, 1, 2, 3, and 1, 2, 3, G3P molecules, so 3 carbons with a phosphate group. Those will then feed into step 5. One thing to note is steps 1 through 4 that we just talked about consume energy. ATP is used during those steps. Steps 5 through 9 actually yield energy. So this is where we get energy back. In step 5, we're starting out with those 2 3 carbon molecules up here. In step 5, a redox reaction will generate NADH. And then in steps 6 through 9, ATP and pyruvate are produced. So notice in steps 5 through 9, every molecule has a 2 in front of it. That is because if we take a look at what went into this, we have those 2 3 carbon molecules. Each one of those 3 carbon molecules will then go through steps 5 through 9. So that is why there's a 2 in front of everything, because 2 G3P molecules will go through all these steps. So each G3P molecule will generate an NADH, but there's 2 G3P molecules right here. So 2 NADHs, so on and so forth. Phosphate is added back in here, 2 of them, 1 for each of those G3P molecules, and we end up with 2 3 carbon compounds with phosphates on either end. In step 6, ADP is phosphorylated to form ATP by substrate level phosphorylation. And then in step 7, some more chemical changes change the structure of our molecule. In step 8, water is a byproduct of that reaction. And then finally in step 9, we have more ATP produced by substrate level phosphorylation, and we end up with 2 3-carbon pyruvate molecules at the end of glycolysis. Again, if you take a more in-depth cell biology class, you will learn exactly what's happening for each of these numbered arrows. I'm not too concerned with it. We're not even going into the chemical reactions. I'm just showing you the intermediates here. Again, the things I want you to pay attention to are what we started with, glucose, what we ended with, the 2 3-carbon pyruvate molecules, and everything that was generated along the way, all the ATP molecules, the water, and the NADH. So this is just showing that last figure with the names of the molecules alongside. So when NAD plus is reduced to NADH, remember reduction is a gain of electrons. So each NADH gains 2 electrons, which means that our organic molecule over here has been oxidized. It has lost electrons. The intermediate here is called 1,3-biphosphoglycerate. Then ADP is phosphorylated to ATP. Our carbon molecule has turned into 3-phosphoglycerate. Some more chemical reactions occur to change the structure, and now we have 2-phosphoglycerate. And then water is lost as a byproduct here. The structure changes slightly. This molecule is called phosphoenolopyruvate. I don't even quite know how to say that. So PEP for short. And then finally, in this last step, we end up with 2 3-carbon pyruvates, and ADP is phosphorylated again to make ATP. In summary, this is probably the most important slide to pay attention to. Glycolysis occurs in the cytoplasm. The reactants of glycolysis are 1 glucose molecule. The products of glycolysis from that 1 glucose molecule are 2 pyruvate molecules, 2 molecules of ATP, and 2 molecules of NADH. The bolded product here is what will go into the next stage, which is pyruvate oxidation. So the 2 pyruvate molecules that are the product of glycolysis will be our reactants in the next stage, which is pyruvate oxidation. That's why they're bold. If you want to go back and look at how these were made, we can go see here. We had the 2 pyruvates there, and then we had the 2 NADH molecules made there. ATP, it looks like we had 4 total made, but notice back in that energy investment phase, we had to use 2 ATP as an energy investment, so the net yield is only 2 molecules of ATP, which is stated here below. Glycolysis produces 2 ATP per molecule of glucose in its net yield. This is because 2 ATP are initially spent during the first half of glycolysis during that energy investment phase, but 4 ATP are then made in the second half, or the energy payoff phase, for a net profit of 2 ATP per glucose. So if you're a little confused still, take a look at this slide and then look at those figures that show glycolysis, and you can actually see where each one of these products is made. The next stage of cellular respiration is the citric acid cycle, which includes pyruvate oxidation. Pyruvate oxidation is kind of a mini step before the molecules get to the actual citric acid cycle, and thankfully it's a pretty short one that's not too complicated. Remember, we ended glycolysis with 2 molecules of pyruvate. Each molecule of pyruvate then will go through pyruvate oxidation. The oxidation of each pyruvate yields acetyl-CoA as its product, carbon dioxide as a byproduct, and more NADH molecules. So our pyruvate, in the first step of pyruvate oxidation, some chemical changes occur and carbon dioxide is given off as a byproduct. In step 2, NAD plus is reduced to NADH again, which means that our carbon molecule over here has been oxidized. In the third step, coenzyme A is added to the carbon, which ends us up with a molecule called acetyl-CoA. Notice that acetyl-CoA only has 2 carbons. This is because one of the original pyruvate carbons was lost as CO2. So there's only 2 carbons, and then those 2 carbons are attached to the coenzyme A from step 3 here. In summary, for pyruvate oxidation, that was a really short one. It occurs in the mitochondria. So we have now shifted from the cytosol to the mitochondria. The reactants in pyruvate oxidation are the 2 pyruvate molecules. Remember, those 2 pyruvate molecules were the products from glycolysis. So the product of one stage of the reaction feed into the next stage of the reaction as the reactants. The products from pyruvate oxidation, we end up with 2 acetyl-CoA molecules, and this is because each molecule of pyruvate has to go through pyruvate oxidation. This is just showing the products from one pyruvate molecule, and we have 2. So one pyruvate will make 1 acetyl-CoA, but the 2 pyruvates that have to go through this will make 2 acetyl-CoAs. Same with everything else. One pyruvate will give off 1 molecule of CO2, but 2 pyruvates have to go through this, so 2 molecules of CO2 will be given off. Finally, 1 molecule of NADH will be reduced, 1 molecule of NAD plus will be reduced to NADH per pyruvate, but 2 pyruvates have to go through pyruvate oxidation, which gives us an end of 2 NADH molecules. So the products of pyruvate oxidation, 2 acetyl-CoA molecules, 2 NADH molecules, and 2 CO2 molecules. That is because we started with 2 pyruvate molecules. Pyruvate oxidation does not produce any ATP, so it produces 0 ATP per glucose. And then 2 of the 6 carbons from the original glucose molecule are released as CO2, 1 from each pyruvate that goes through pyruvate oxidation. So that was pyruvate oxidation. It was a nice and somewhat easy one to understand. Now we're going to go to the second part of stage 2, which is the citric acid cycle or the Krebs cycle. The citric acid cycle completes the energy yielding oxidation of organic molecules, and it's often represented with a circle, and each circle represents 1 turn of the citric acid cycle. So for each turn of the citric acid cycle, you'll get 2 carbons from acetyl-CoA being added to the cycle. 2 carbon dioxides are released, which we see right here. And then 3 NADH and 1 FADH2 are produced. If we go ahead and count up the NADH, we see that this tells us that 3 NADH are produced here, and then 1 FADH2 is produced there. That's where those numbers are come from. And then an ADP is phosphorylated again to make ATP. This is a very simplified image of the citric acid cycle. The citric acid cycle will turn once for each acetyl-CoA molecule added. Remember, at the end of pyruvate oxidation, we have 2 acetyl-CoA molecules because 2 pyruvate molecules went into pyruvate oxidation. So 2 acetyl-CoA molecules means that the citric acid cycle will turn twice. These numbers are just for 1 single turn around the citric acid cycle. These arrows just represent the things that are happening. Coenzyme A breaks off here. Carbon dioxide is released here. 3 NAD pluses are reduced to NADH here. They gain electrons. ADP is phosphorylated to ATP here. And then FAD is reduced to FADH2 plus there. A reminder again, the pyruvate oxidation and the citric acid cycle do take place in the mitochondria. And the products of pyruvate oxidation, the 2 acetyl-CoA molecules, feed directly into this citric acid cycle. Now we're going to take a more in-depth look at the citric acid cycle. This last image here made it look somewhat manageable. Now we're going to take a look at all of the individual steps, which are a lot. Again, I don't expect you to memorize molecule names, but I do want you to understand where these products here are coming from. So in step 1, we start out with our acetyl-CoA. So 2 carbons from our acetyl-CoA, notice it has 1, 2 carbons, enter the cycle, and a 6-carbon molecule citrate or citrate forms. Notice right here, before those 2 carbons even enter the cycle, the coenzyme A breaks off of acetyl-CoA. So now we just have 2 carbons entering the cycle. Those 2 carbons are going to combine with oxaloacetate and form a 6-carbon molecule, which is citrate. This is where the citric acid cycle gets its name. In steps 2, which is right here, and then 3, which is right there, NADH, ATP, and CO2 are generated during a series of redox reactions. So in step 2, there's a few things that happen to turn citrate into alpha ketoglutarate, which is a 1, 2, 3, 4, 5 carbon molecule, but citrate is a 6-carbon molecule. That means we have to lose a carbon somewhere along the way, and we can see right here we lost the carbon as CO2. So that is why we only have a 5-carbon molecule. And somewhere along this stage as well, NAD+ will be reduced to NADH. We now have alpha ketoglutarate. Alpha ketoglutarate is a 5-carbon molecule, but notice our next carbon molecule is a 4-carbon molecule, succinate. This means alpha ketoglutarate is going to lose a carbon, and we see that right here with CO2 leaving the cycle. So there's 2 points where CO2 leaves the cycle, here and here. In step 3, NAD+ is again reduced to NADH. So that happened up here for 1 NADH molecule. Now we have 2 NADH molecules because that happened again. And remember, every time NAD+ is reduced to NADH, a hydrogen ion is released. Those hydrogen ions become important later, so just kind of keep that in the back of your mind. We have those 2 NADHs so far, and then in stage 3, ADP is phosphorylated to form ATP. In steps 4, 5, and 6, we see more electron carriers reduced and water added. So in steps 4 through 6, further redox reactions generate FADH2 and then more NADH up here. FAD is reduced to FADH just once, and succinate becomes fumarate. Water is added in right here, and then malate becomes oxaloacetate. As malate becomes oxaloacetate, NAD+ is reduced to NADH. So we can see in total we have 1, 2, 3 NADHs produced per turn of the citric acid cycle, 1 FADH2 produced, and 1 ADP phosphorylated to ATP. After stage 6 occurs, we end up with oxaloacetate again, which then just feeds right back into the Krebs cycle, and it will combine with the next 2 carbons that enter the cycle. So the citric acid cycle is circular and keeps going around recycling the oxaloacetate every time. In summary, the citric acid cycle occurs in the mitochondria. The reactants are those 2 acetyl-CoA molecules, which we see right here. Remember, pyruvate oxidation produced 2 of them, so 2 acetyl-CoAs have to go through the Krebs cycle or the citric acid cycle, which means there will be 2 total turns of this cycle. This image here just represents 1 turn and the products from 1 turn of the cycle. The cycle has to go around twice. So the products are the products of 2 turns of the citric acid cycle. So we're going to double all of the products from 1. So 1 turn will give us 1, 2, 3 NADH. Double that. That means we have 6 NADH. 1 turn will give us 1 FADH2. We need to double that. So 2 FADH2. And then 1 turn will give us 1 ATP. We then double that to get 2 ATP. And we see that shown in the product. So we have 6 NADH molecules, 2 FADH2 molecules, 4 CO2 molecules and 2 ATP molecules. We didn't look at the CO2, so we'll look at that really fast. From one cycle, we can see that we have 2 carbon dioxides leaving the cycle. So from 2 cycles, there would be 4, which matches up with these products here. The citric acid cycle produces 2 ATP per initial molecule of glucose, just because each acetyl-CoA that goes through the cycle produces 1 ATP. So per 1 molecule of glucose, the citric acid cycle will turn twice because 2 acetyl-CoAs enter that cycle. The remaining 4 carbons from the original glucose molecule are released as CO2, 2 from each acetyl-CoA. So each time the cycle turns again, 2 carbon dioxides are released. This is the citric acid cycle summary. The bold products in this one are the NADH and the FADH2 molecules, because in stage 3, it's all the electron carriers that become important. So each stage so far, glycolysis, pyruvate oxidation, and the citric acid cycle, all of them have either produced NADH or FADH2 molecules. Those molecules, those electron carriers, become important as we start to talk about oxidative phosphorylation, which is stage 3. Stage 3 is where most of the ATP production occurs, and it's called oxidative phosphorylation in total. It has 2 parts. The first part is the electron transport chain. The second part is chemiosmosis. We're going to talk about the electron transport chain first. So this part in this little figure. In the mitochondria, because oxidative phosphorylation also occurs in the mitochondria, electrons from our electron carrier molecules, NADH and FADH2, are passed down the electron transport chain to oxygen, which is the final electron acceptor. Oxygen then picks up some hydrogen ions to form water, which is why water is a byproduct of cellular respiration. We're going to take a quick look at the passage of those electrons from NADH and FADH2. I know there's a lot going on in this figure, but I'll try and simplify it as much as possible. So we have NADH here carrying its 2 electrons, and then FADH2 here carrying its 2 electrons. NADH drops its electrons off in the first electron carrier complex right here. FADH kind of skips this first electron carrier, and it drops its electrons off at electron carrier 2, which is right here. The yellow arrows just represent the path of electrons. So NADH's electrons flow through electron carrier 1 to this mobile carrier Q to electron carrier 3, another mobile carrier that just shuttles the electrons, and then finally to electron complex 4. The electron carrier complexes 1, 2, 3, and 4. At each one, the electron kind of falls down an energy staircase as it's accepted. So electrons shuttled from glycolysis, pyruvate oxidation, and the citric acid cycle are delivered to the electron transport chain. As the electrons get passed between the electron carriers, those carriers use some of the energy generated to pump hydrogen ions, H+ ions, across the membrane. So we can see here, those green arrows represent the pumping of hydrogen ions. The electron carrier complexes 1, 3, and 4 will pump those hydrogen ions out. Electron carrier complex 2 does not. And again, the mobile electron carriers Q and site C right here just shuffle electrons between the different electron carriers. Once NADH drops its electrons here, they're shuffled along this path until they end up with oxygen, which is the final electron acceptor. Oxygen accepts those electrons and then joins with 2 H+ ions to produce water. Remember, think of these electron carriers as parts of the electron transport chain, even though they're arranged in the mitochondrial membrane. Each one of these purple dots represents one of those electron carriers that we just saw. So electron complex 1, 2, 3, 4. Those are represented by these purple dots here. As those electrons are shuttled between them, energy is given off, which is used to synthesize ATP, but it's kind of a complicated way how it synthesizes that ATP. And then the final electron acceptor down here is oxygen, which joins with the electrons and two hydrogen ions or H+ ions to create H2O. We'll talk about how specifically this energy is used to synthesize ATP when we talk about chemiosmosis, which we'll get to on the next slide. Energy released by these redox reactions is used to pump hydrogen ions into the inner membrane space, which we already talked about. Those are those green arrows right here. So the energy from the electrons falling down the staircase in between these electron complex molecules are providing energy, or is providing energy, for hydrogen H+ ions to be pumped across the membrane by electron complexes 1, 3, and 4. This hydrogen ion gradient is really important when we get to chemiosmosis, because the hydrogen ion gradient drives H+ ions back through the enzyme complex ATP synthase, shown right here. ATP synthase is located in the inner membrane, and as those hydrogen ions pass back through ATP synthase down their concentration gradient, they spin the rotor of ATP synthase kind of like a wheel, which gives the energy for ATP synthase to phosphorylate ADP to ATP. So chemiosmosis is the part that's occurring over here with ATP synthase. The electron transport chain serves to take all the energy from those electrons and use it to pump hydrogen ions across the membrane. So I know that was a lot. This image here is a little bit simplified. It doesn't have all that text, and I'm going to talk through this one because hopefully it's a little easier to conceptualize everything. So the yellow arrows again represent electron flow. NADH and FADH2 drop their electrons off at different locations in the oxidative phosphorylation, the electron transport chain. NADH drops its electrons off at complex 1. Once it drops its electrons off, it is oxidized back to NAD+, because remember, oxidation is lost. It has now lost its electrons. Those electrons that it dropped off at complex 1 are shuttled along this pathway through complex 3 and then through complex 4. Each one of those complexes, the electrons are falling down the staircase as they get passed between them, which gives off energy. This energy is used to pump hydrogen ions across the membrane against their concentration gradient. So the concentration of hydrogen ions is higher outside the membrane. The hydrogen ions from inside the membrane are being pumped against their concentration gradient from low to high. So energy is needed to do this. The falling of the electrons provides that energy. And so each complex will pump hydrogen ions across the membrane as the electrons fall down that staircase, ultimately being accepted by oxygen and then combining with two hydrogen ions to form water. FADH2 drops its electrons off at complex 2, so it skips complex 1. So the ability of FADH2 to contribute to ATP synthesis is a little bit less than NADH, just because it misses this chance to transport hydrogen ions across the membrane. However, it funnels them to 2, which they then follow this path, passing through complex 3 and complex 4, which them falling down the staircase provides the energy to pump hydrogen ions across the membrane until those electrons are finally accepted by oxygen to form water. Once FADH2 has dropped off its electrons, it is oxidized to FAD. It no longer has electrons. Now we get to chemiosmosis. The important part for chemiosmosis is this hydrogen ion gradient that has been created by the active transport of hydrogen ions against their concentration gradient. There are a lot more hydrogen ions out here than inside the membrane. So where do they want to go? According to the rules of simple diffusion, they want to go to where there's a lower concentration. They want to move from high concentration to low concentration. And they do this through ATP synthase. each time a hydrogen ion moves through ATP synthase, it spins this rotor. You can think of it kind of like a wheel, like a spinning wheel. The spinning of that rotor then provides the energy to phosphorylate ADP to ATP. So I have a video that summarizes this whole process of oxidative phosphorylation.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So we're going to pause that video there and go back to our PowerPoint. So that video did an excellent summary of the electron transport chain and chemiosmosis. Interestingly, they talked about chemiosmosis first and then the electron transport chain second, but they did a good job of explaining everything in even more detail than we went into in this lecture. So hopefully you found that video helpful. In summary, oxidative phosphorylation occurs in the mitochondria. The main reactants part of this are electrons from the electron carriers, which are the NADHs and the FADH2s. NADH has been produced in glycolysis, in pyruvate oxidation, and in the citric acid cycle, and FADH2 was produced in the citric acid cycle. So all of those electron carriers donate their electrons to the electron transport chain during oxidative phosphorylation. The products of the electron transport chain and of oxidative phosphorylation in general are water and ATP. Oxidative phosphorylation is the heavy hitter of cellular respiration. It produces 28, roughly, molecules of ATP per molecule of glucose, which is significantly more than any of the steps we've looked at thus far. All of the NADHs and FADH2s from the previous phases drop off their electrons into the electron transport chain, where their energy of them falling down the staircase is used to pump H+ ions across the membrane. The electrons are picked up at the end of the electron transport chain by oxygen, which then becomes the product water. The H+ ions diffuse back through the membrane through ATP synthase. So their movement is harnessed by ATP synthase to phosphorylate ADP into ATP. And 28 molecules of ATP per glucose are made. A quick checkpoint question on oxidative phosphorylation. What effect do you think an absence of oxygen, O2, would have on the process of oxidative phosphorylation? take a second and think about this, and think about if oxidative phosphorylation would be possible if you didn't have oxygen. So without oxygen to pull electrons down the electron transport chain, the energy stored in NADH and FADH2 could not be harnessed for ATP synthesis. This is why we always have to constantly be breathing in oxygen, and why very quickly without oxygen you will die, because all of the cells in your body need the oxygen that you're breathing in to be the final electron acceptor in oxidative phosphorylation. If you don't have it, it won't pull the electrons down strong enough, and ATP synthesis won't occur. So in summary, this is our last slide for this video. Cellular respiration occurs in 3 distinct phases. Stage 1 is glycolysis. Stage 2 is pyruvate oxidation and the citric acid cycle. And then stage 3 is oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. We're going to quickly go through each of these. Glycolysis starts out with the reactant glucose, and it produces the product of 2 pyruvate molecules. 2 ATP are also produced by substrate level phosphorylation during glycolysis, and 2 NADH are produced in glycolysis, which get sent to the electron transport chain. In pyruvate oxidation, 2 NADH are produced that also get sent to the electron transport chain, and then CO2 is given off as a byproduct. In pyruvate oxidation, the 2 pyruvate molecules from glycolysis are the reactants, and then 2 acetyl-CoA molecules are the product. Those 2 acetyl-CoA molecules feed into the citric acid cycle. They're the reactants in the citric acid cycle. The citric acid cycle gives off 2 ATP by substrate level phosphorylation, and then 6 NADH and 2 FADH2, which carry their electrons to the electron transport chain. In oxidative phosphorylation, the electron transport chain, all of these electrons are dropped off, and then they fall down that energy staircase, releasing energy to pump H+ across the membrane. The final electron acceptor is oxygen, which accepts the electrons and then 2 hydrogen ions to become H2O. And then that H+ ion gradient across the membrane pumps ATP synthase, which generates about 28 ATP by oxidative phosphorylation. So in total, the maximum amount of ATP per glucose is 2 + 2 + 28, which gives us about 32 ATP. This can vary, but for the most part, it is 32 ATP. Okay, I'm going to end this video here, and I'll see you in the next video where we're going to talk about fermentation, which is an anaerobic harvesting of energy.